Thank you. So, hello everyone. My name is Sandra Rodríguez Quintana. I'm a postdoctoral research associate at Community Environment and Policy. And today I will be presenting some of the results or a special project that we have, an ongoing project, right, in Guatemala. And it's titled Infant Exposures to Chemicals and Pathogens in the Lake Atitlán Watershed, Guatemala, and the Development of Effective Interventions. So over 149 million children under the age of 5 are stunted worldwide, and Guatemala ranks 6th and has the highest stunting prevalence in the Americas. So what's stunting, right? So stunting would be the inadequate linear growth defined by the height that is more than 2 standard deviations below the child growth standard medians. And there could be multiple factors that contribute to the development of stunting. That could be poor maternal health, recurrent infections, inadequate feeding practices, and all this can be tied on a distal end to poverty. Why stunting is concerning is because it can have long-term consequences on health outcomes. It could be physical, cognitive development, educational attainment, and also affects productivity as people grow as adults, right? So here we can see the World Health Organization conceptual framework that includes all of these factors that I mentioned and others. We can see, for example, that they mention inadequate breastfeeding. So we know that breast milk is the optimal nutrition for children. So when we have inadequate, it might be contributing to stunting. But also we know that breastfeeding could be a protective factor for stunting. Also, there's another factor that I want to focus on, that is the water, sanitation, and environment, that this is one of that we are focusing with this project. So Guatemala is the second largest indigenous population in Latin America. We focus on this project in the Lake Atitlán specifically. These communities, the economy is based around the lake for fishing, economy, for food, agriculture. And there's previous studies from our lab as well that have report that there's some contamination around the lake, in the Lake Atitlán, in the water. This could be from metals, metalloids, pesticides, but also pathogens. So around the Lake Atitlán, over 70% of mothers breastfeed. So that's an important point that we mentioned, right, for as a protective factor for stunting. So that's kind of the focus with this project. So the objectives are to assess the concentrations of the toxic metals, metalloids, OCPs in the breast milk. With this, examine associations with infant stunting in this population, and this to provide some evidence to inform public health interventions. So as part of the methods with the University of Arizona, a local university, Universidad del Valle de Guatemala, and also an NGO in Guatemala, so we conducted this pilot study in 2022 when it started. As I said, it's a continuous. And we focus on 4 communities around the lake. We focus in these communities because of the prevalence of stunting, some of them even more, up to 60% of prevalence of stunting, and also for the accessibility for the research team. The study population was an 80 mother-infants diet. We were focusing on children, on infants from 3 to 6 months of age that we will be exclusively breastfed. And we also collected data on length to assess stunting, right? Okay. So after we did all the analysis and the results, I'm going to show you quickly, right, our results. So specifically for stunting, from these 4 communities, 2 of the communities show higher prevalence of stunting in the infant population. We can see Panajachel and Santiago Atitlán. The purple line is the definition for stunting based on the WHO, and the red one would be for severe stunting. So when we compare these 2 communities, they have higher prevalence compared to the other 2. However, the optimal thing would be that all the infants would be above 0, okay? And when we analyze the concentrations of the metals and metalloids, we found an association with the concentration of 4 metals, metalloids, and with the prevalence of stunting. So those were arsenic, barium, beryllium, and lead. And when we analyzed the concentrations per communities, we found that the concentrations were higher on the communities with the higher prevalence of stunting, the ones that we mentioned before, specifically Pathogens. And we can see, like for arsenic and lead, that the concentrations were above of what is recommended by the WHO guidelines for breast milk concentrations. So for OCPs, when we conducted the analysis, we didn't find like any association with the prevalence of stunting. However, there were concentrations in the breast milk samples. The only one that was marginally associated with the prevalence of stunting was for DDT. And in addition to do all this analysis in the breast milk samples, we know that there are other factors that might be contributing to stunting. We also collected some drinking water samples. And as I said, this is an ongoing project. We have here our PhD student and part of our team, Yunoen Soto Fernandez, who is focused more on the microbiological side and the drinking water. So we found, for example, that there were levels of arsenic that were elevated on the water. And also, she's conducting some analysis and also found like higher proportion or percentage of E. coli, right, in this water sample. So this has like some ideas that we're trying to connect and see what might be some interactions or factors that are contributing to stunting. So for example, the environmental enteric dysfunction. So if these infants are exposed to pathogens and they ingest the microbes, there could be some alterations in the small intestine that can affect the absorption and utilization of nutrients, and this also can contribute to impairment growth. So, some key findings for the study. Maternal exposure, there's maternal exposure to toxic metals, metallos, and OCPs in this part of Guatemala, in the Lake Atitlán. We saw that there's some associations with reduced growth, and that this pollution threatens infant development. So there's need for safe drinking water based on some of the results that we have, and also for implementation of WASH interventions. And as I mentioned, breastfeeding, optimal nutrition for infants, so to reinforce that it still protects infants. So next steps, continuing partnering with local organizations and conducting a longitudinal study where we have like pre and post water filtration so we can test for chemicals and also pathogens. And the idea that we have now is like the sequence for pathogen identification so we can kind of modeling some interactions among these toxic chemicals and also the pathogens and how it affects child development. That in the end, that's the main reason, right? So that all children can thrive and have a healthy development. So yeah, thank you for your attention.